Welcome to ICBAS Biomedical School of Sciences International Webinar Series. It is my pleasure to introduce the speaker of today's webinar, Nick Proudfoot. Nick has been a great contributor to ICBAS PhD programs, as he has hosted in his lab and co-supervised several of our PhD students. He acts as a member of the External Advisory Board of the MC Biology PhD program, and he has lectured for this program as well as for the GABA PhD program. And I want to thank you for your valuable contribution to our school. Nick made outstanding contributions to the field of molecular biology and RNA, as most of you know, with an impressive publication list. And he has been an inspiration for many students and scientists throughout the world. As you can see, because some of your former members of your lab are here today, just to listen to you. He studied biochemistry, and then he did a PhD in molecular biology at LMB in Cambridge, under George Brownlee's supervision in Fred Sanger's section of LMB. And some of you have heard, the students have heard about Sanger sequencing. This was at a time when Fred Sanger and George Brownlee worked out various techniques to sequence RNA and DNA, and when restriction enzymes were just about to become available. So exciting times. At the time, Nick discovered the poly-A signals that marks messenger RNA molecules for transcription termination. And everybody is hearing about mRNA these days, so Nick is an expert on that. And Nick proposed that this signal is really important for mRNA biogenesis. He then moved to Tom Maniati's lab, and some of you have heard about Maniati's as well, in Caltech and Harvard, where he cloned and sequenced several first genes from mRNA isolations. He then went to the Sir William Dunn School of Pathology at the University of Oxford. He became a fellow and tutor for Brasenose College, where he taught biochemistry and molecular biology. Nick has won multiple awards and distinctions, including being elected as an EMBO member. He's a fellow of the Royal Society and a Wellcome Trust investigator, and has served on several scientific boards of various research institutes. He's now a professor of molecular biology and runs a joyful and very productive lab. Nick has come to the IBMC ICBAS in various occasions, but this time I'm very pleased that he accepted to be here to give this seminar at ICBAS. And it's a pleasure to host you at virtual ICBAS. Welcome, Nick, and the stage is yours.
Fine. Can you hear me, Alex? Alex, can you hear me?
Yes, Nick, I'm here.
Okay, so I shall begin, right?
Yes.
Okay. So thank you so much.
40 minutes.
I know. Thank you so much, Alex, for your introduction. So my remit for this talk is to give you a little bit of history, probably about half the talk, and then I'll give you a very potted kind of description of the kind of work we're doing at the moment, which of course is quite complex, and so it'll be very simple cartoons rather than any data. Okay, so yes, the other realization I came with producing this talk is that I have been doing molecular biology a very long time, and I actually started in the lab as a grad student in '73, and it's getting very close to 50 years. So I suppose witnessing the 50-year gene revolution from genes and mRNA to genomes and transcriptomes is perhaps not too inaccurate to describe my career, basically. So how did it begin? Okay, so I started as a grad student in LMB Cambridge in 1973. Can you hear me? Yeah. Yes. And I was lucky to get a place in Fred Sanger's section, and I was working supervised by George Brownlee. And it was an amazing time to be doing molecular biology because this was really the very beginning. But the LMB in Cambridge was probably the kind of the place where the most molecular biology was happening. And this is really indicated by the incredible list of names, all of which on this list essentially obtained Nobel Prizes, which is pretty amazing. So actually, when I joined, there were 6 or 7 current Nobel laureates, and then subsequently a lot of other people I knew when I was at LMB Cambridge got Nobel Prizes. So yes, it was clearly, there was clearly no excuse but to not do some reasonable science, because I was surrounded by so many amazing people. But the thing about molecular biology when I began was that we really knew nothing and we could really do nothing. And that was the remarkable feature of the field at that time, because basically when I came to the lab, we didn't know anything about how genes were arranged in eukaryotic genomes. No genes had been purified. There are a few messenger RNAs that were purified rather crudely, but they were there. In fact, I used them. We didn't know anything about gene structure. We didn't know about introns and exons. We didn't know how DNA was arranged in the nucleus. We had very rudimentary ideas about chromatin. Nucleosomes hadn't been discovered. I mean, the list just goes on. It's extraordinary, really. But that's what we didn't know. But of course, the reason we didn't know anything is we couldn't do anything. And you just look at what we couldn't do when I came to the lab. But we didn't have restriction enzymes, reverse transcriptase. There was no PCR, of course. But probably more significantly than that, there was no plasticware. We didn't have Eppendorf tubes. We couldn't pipette. We didn't have pipettemen. So what we had to do when I came to the lab, we had to make our own calibrated glass capillaries for 1 and 2 and 3 and 4 microliters. And so you use the same capillary over and over again because it took you half a day just to calibrate it. So there was a bit of a problem with cross-contamination, clearly. We didn't have gels, agarose, acrylamide gels. The chemicals were there, but we didn't know about using slab gels at all. Obviously, there was no blotting because there was no gels to blot anyway. We didn't know how to sequence DNA. A bit of RNA sequencing had been developed, but DNA sequencing was just beginning. And of course, we didn't have gene cloning. That's why we didn't have any genes. So, it was amazing. We didn't have any of the kind of technologies that are now all taken for granted. And, but of course, the point about this position was that if you could get anything to work, then it tended to be quite a fundamental discovery that you could make because nobody knew anything. And so it was sort of the pros and cons to not being able to do everything. I mean, nowadays, there's so many techniques around to do everything that it's kind of hard to find something different from everybody else to do because there's so much technology around. When I wrote my thesis in 1976, this was basically our view of gene expression, which is pretty amazing, really. So we knew that DNA was in the nucleus. We knew that genes were copied into some sort of weird transcript. We didn't know what the transcript was. We called it heterogeneous nuclear RNA. We knew that it had a poly-A tail at its end, but that's about all we knew about it. And then somehow this very heterogeneous large RNA was converted into a messenger RNA. Again, we didn't exactly know how. And transported out of the nucleus to the cytoplasm. Again, the nuclear pore complex hadn't been discovered, although there were blobs on the outside of the nucleus that people knew might be something to do with export. Then, of course, once in the cytoplasm, the messenger RNA was translated. So quite a bit about the basic ribosome was known, but not the details. And of course, how the RNA is degraded after it's been translated for a while was poorly understood. So when I joined the lab, there weren't any genes cloned. In fact, there was no cloning. We didn't know how to clone genes, but there were a few messenger RNAs available, particularly globin messenger RNA. So what we could do was we could isolate messenger RNA from weird tissues which made a lot of one particular protein. So they had therefore quite a lot of that messenger RNA. So globin was a good case. Also, immunoglobulin messenger RNA, you could get that out of B cell lines, myeloma cells. But it was very, yes, also sea urchins were a good source of histone messenger RNA, it turned out, because sea urchins have a period in their development when they just divide their cells incredibly fast, and so they make a lot of histones. But that's kind of where we stood. There were very few messenger RNAs around, no genes. So how on earth could you do any molecular biology? Good question. Well, my project when I joined the lab was to see if I could copy these few messenger RNA, and it was basically globin messenger RNA, see if I could copy it into some sort of DNA copy. That was the idea we had, but of course we didn't have reverse transcriptase, and so what we did was we knew that you could use E. coli DNA polymerase, and if you gave it a buffer with manganese, then you could get it to copy an RNA template. instead of its regular DNA template. And so you can, in fact, make a short cDNA using a simple reaction. And this cDNA, you can make it radioactive because we would use alpha labeled nucleotides. They were available, although we had to make them ourselves, I would say. We couldn't just order alpha P32 labeled triphosphates. We had to synthesize them in the lab, which was a rather radioactive experience. And so then these short P32 labeled cDNAs, at the time we were sequencing RNA a bit, and the way you sequence RNA was you broke it into little pieces and you fragmented the pieces in 2 dimensions on chromatography to give you an RNA fingerprint. So we tried the same thing with DNA and made a DNA fingerprint, which is this picture here. So this is basically a 2-dimensional fractionation of digested cDNA, cut not with a restriction enzyme, but with an endonuclease that we extracted from E. coli that's called endonuclease 4. It's fairly non-specific, but it seems to like to cut mainly at Cs for various reasons. So we had these radioactive spots, as we called them, and we isolated the spots, and then we chewed the spots from one end, sequentially, to give a picture that's quite pleasing to the eye, and it's called the wandering spot fractionation, because basically all of these fragments are different from the next one by 1 nucleotide. And then we realized that the mobility change from this spot to that spot was characteristic of which base was different between the 2 of them. And so from that, we could read the sequence, although not very effectively. It was often a bit of a, it wasn't very precise, but you could get a sequence. And so I managed to do this basically. And because the endonuclease 4 fingerprint was a partial digest, I had overlapping spots. And so I could work out the complete cDNA sequence, starting with so I was priming the messenger RNA from the poly A tail with oligo Ts. So the first part of the sequence was a run of Ts. And then I had this cDNA. And that's what I did. And I did this on it turns out 6 different messenger RNAs became available. So I did all 6 of them, and found this common sequence, AAU-AAA, about 20 bases away from the poly A tail. So obviously I'm copying the cDNA back into RNA here. And so basically that was what I did for my thesis. In fact, my thesis was basically the sequence of about 300 nucleotides. In fact, I knew every single base of the sequence I deduced for my doctoral thesis, but it was kind of an interesting sequence because it was 6 different cDNAs that all had the same AAU-AA3. And so that then led myself and George Brownlee, my supervisor, to write this sort of little letter to Nature. And we rather sort of ambitiously proposed that maybe AAU-A3 was something to do with the poly A tail, because it's quite near the poly A tail at the end of the message. And that turned out to be right, because actually with genomics and the vast amount of sequencing data we now have, we know that all messenger RNAs pretty much end up with this sequence AAU-A3 that's just before the poly A tail. So it was a very lucky break, and it was in a sense, as I said, it's like early bioinformatics. It was quite a simple bioinformatic task because there were only six sequences to look at, and in fact, I found the sequence by just looking at by eye. We didn't have any sort of techniques for sort of using computers to search for sequences. We just use our eyes. But it was sort of bioinformatics in a sense. Okay, so and yes, the other thing was that we called these sequences near the poly A tail of the messenger RNA non-coding, because they didn't directly code for the protein sequence. We know what the protein sequence for globin was, but we knew that the sequence didn't match that globin amino acid sequence because obviously we knew the genetic code. And so we figured this must be some sort of extra bit of sequence at the end of the messenger RNA. So we called it non-coding RNA. And in fact, this was probably the first time this term was used. And annoyingly, people in the States switched the word non-coding to untranslated, which you know, they never asked my permission, even though we were the first people to describe it. So going on, we didn't really prove that this sequence was important directly, but happily there was a mutant in a globin gene obtained from a patient with alpha thalassemia, which is basically when you don't make any alpha globin or don't make enough alpha globin. And this mutation converted the AATA3 to AAT-AAG. And myself, together with a guy in the clinic, in the medical lab that was working on alpha thalassemia called Doug Higgs, the two of us, together with a few other people, worked out that in fact this mutation prevented the alpha 2 globin messenger RNA from being formed, essentially because it wasn't chopping at the 3' end and adding a poly A tail. So the RNA is very unstable. And in fact, we also noticed that this mutation had two effects. You didn't make any alpha 2 globin messenger RNA, but also you made a lot less of the downstream alpha 1 globin gene. And we now know that's because actually, if you don't terminate transcription over alpha 2, you then run into alpha 1 by a process that we call transcriptional interference, and this blocks the expression of alpha 1. And so it was actually an experiment that led to a lot of new concepts, like the requirement of the poly A signal and the idea that there might be a connection with transcription that resulted in transcriptional interference. Going on, yeah. So we did these rather hideous experiments that we called nuclear run-on analysis, which basically involved taking the cells that we transfected these alpha globin constructs, either the wild type alpha globin gene or the one with the poly A mutation. So we transfected these into our Hela cells, which were the standard cell lines we used in the lab. And then we stuck a lot of we isolated nuclei from these cells, and then we incubated the nuclei with really a large amount of alpha P32 UTP, about a half a millicurie of it usually. And we got these very sort of just above background sort of RNA signals, which we thought were nascent transcripts. And so we got a nascent transcript over the body of the alpha 2 globin gene, but nothing much downstream. It was just background. But then the mutant gave you equal amounts of signal over the body of the gene and downstream. So that really proved that the poly A mutation was indeed affecting termination as well as the processing of the transcript at the 3' end. And that was a mammalian experiment, but actually my lab then switched for a while into yeast, because it's a much neater genetic system and you get much cleaner data if you do things right. And so looking at a similar experiment in S. cerevisiae, we had a CYC1 gene with a poly A signal mutation. And so normally the gene makes a nice messenger RNA. This is a northern blot. And so if you look at the mutant that doesn't have the poly A signal, you make very little of the message. But then if you do the run-on analysis, the same idea that I described in the Hela cells, you incubate the yeast with a large amount of radioactive UTP, but it's just a lot cleaner. And so you get a very nice set of signals over the body of the gene, probes 1, 2, and 3, but then it stops abruptly after probe 3. But then in the mutant that doesn't have the poly A signal, it runs through. So that was a very beautiful demonstration that really the poly A signal is required both to make the message and also to prevent to cause the polymerase to terminate transcription. If you don't have that poly A signal, you don't terminate anymore. Right, so then going on, so we wanted to find more about what these signals might be. We knew that AAUA3 was part of the poly A signal, but maybe there are other parts as well nearby. So we found there was an element downstream of the AAUA3 that was important for the poly A signal to work, a GU-rich region. And then a very talented graduate student joined my lab and found there was another signal upstream of the AAUA3, the so-called upstream sequence element, and you'll recognize the name Alessandra Moreira. So she was in my lab a little while ago as a graduate student. So going on then to the next part of this sort of history of poly A signals. So not my lab, but the labs of Jim Manley and Walter Keller worked out the biochemistry and found the protein factors that recognize the poly A signal and cleave the transcript. And so these proteins with very easy to forget sort of initials like CPSF and CF1 and CSTF, which basically bind the poly A signal and then cleave the transcript at this red arrow, and then the poly A tail is added following the initial cleavage. And so that's basically the biochemistry of cleavage and polyadenylation. But how does this connect to transcriptional termination? Oh, hang on, before we get to termination. So I showed you just a few factors here, but in fact, when you look at all the factors and do the proper proteomics, there's a load of factors involved in cleavage polyadenylation. In mammals, there are probably, well, and this is quite an old slide, goes back to 2004, but even today, there are probably 50 or so polypeptides involved in mammalian cleavage and polyadenylation, and there's probably more than that involved in yeast cleavage and polyadenylation, and obviously coupled termination too. So it's very complex. And why do you have such great complexity of factors when all you need to do is chop the transcripts and add a poly A tail at the end? And this led me to a favorite cartoon figure that I often show in my history lectures, which was a cartoon produced by Marv Wickens, who's another great expert in poly A signals. And he had this cartoon of a gorilla hanging onto a vine. And the question is, why do you need an enormous gorilla to chop the vine and add the poly A tail? And there's an extra amusement to this cartoon or this picture, because Marv Wickens is a very big guy, and for a while he was extremely hairy. He had a long brown beard, and so we all thought this was closer to Marv Wickens than he would have liked, perhaps. So going on, so the poly-A signals required for cleavage and polyadenylation, but how does it relate to termination? And so the idea is that the RNA polymerase II, when it transcribes along the gene, this is this wiggly double-strand DNA. So here's the RNA polymerase II transcription bubble, and this part of RNA polymerase II is the CTD, which is the C-terminus of the largest subunit of RNA polymerase II, which recruits the RNA processing factors to the polymerase as the polymerase makes the transcript. And these factors then act on the transcript as it's being made co-transcriptionally. And so here are the cleavage poly-A factors bound to CTD, cleaving the poly-A signal to release the messenger RNA with a poly-A tail on it. But then what we realize is that the polymerase keeps on going. It'll continue to transcribe even if you chopped away the messenger RNA. But then the way it terminates is that onto this 5' end generated by cleaving at the poly-A site, you now attract an exonuclease, 5' to 3' exonuclease called XRN2 in mammals or rat1 in yeast. The annoying thing is yeast gene names are always, or protein names are often different to the mammalian equivalents. But anyway, by chewing a transcript, this actually promotes the release of the polymerase from the DNA template. And so we think the way termination works is that you chop away the message, and then the transcript is still being made by the polymerase. If it's degraded by this exonuclease, and that exonuclease catches up with the polymerase, that promotes transcriptional termination. Probably if the polymerase slows down, pauses, that helps the process. And we published this in 2004. Our paper was a mammalian sort of analysis, and then the side-by-side paper was from Steve Borotowski on yeast. So going on, okay, so that's pretty much the history. Now I'm going to talk about more recent stuff my lab's been involved in. It really what my lab for the last decade has focused on is this paradox, which is that we have 20 to 30,000 protein coding genes in the human genome, which is about 1 to 5% of the genome length. So what's all the rest of the genome doing? And of course, anyway, 20 to 30,000 protein coding genes isn't nearly enough to account for protein diversity. But we now know that splicing and alternative splicing generates quite a bit of this extra diversity. Rather than the rest of the genome not being active and not doing anything, the big surprise has come from recent sort of transcriptomic analysis, which is that basically a lot of the extra genome is still transcribed, but it makes non-coding transcripts or long non-coding RNA. So this goes back to the non-coding sequence I found at the 3' end of messenger RNAs. It turns out there's a lot of non-coding RNA, and mainly it's not the end of a messenger RNA. It's these extra transcripts that are made between protein coding genes. And of course, protein coding genes themselves are highly intronic. So remember that many protein coding genes will be 95% intron. So really, there's an incredible degree of transcriptional wastage, if you like. We make loads of transcripts that don't code for any particular protein. And the other bizarre thing is that transcription, we know, is a bit dangerous. So if you make transcripts, there's always a chance the transcript will get caught up and re-anneal with a DNA duplex to form something we call an R loop. R loops are quite mutagenic, and furthermore, R loops cause the polymerase to get stuck. And if the RNA polymerase gets stuck, and if this happens during the S phase of the cell cycle, then the stuck polymerase, RNA polymerase, can interact with the replication fork to give you a collision, and then you get double-strand breaks. And so, altogether, transcription, you don't want to have a lot of extra transcripts, but that's exactly what we seem to have. So it's a paradox. So basically, my lab switched at this point from looking at single genes, mainly just globin genes, to be honest, to looking at the whole genome and the whole transcriptome. And really, this switch in my lab, I found personally to be a bit difficult, because I tended to just like to look at individual genes and their specific sequence. And so the concept of looking at the whole transcriptome requires the analysis of big data, and big data requires computation, and it particularly requires a breed of scientists called a bioinformatician, which I'm clearly not. And so really, the last 10 years of my lab has been very much down to, one, developing techniques which allow us to generate these genome-wide transcript profiles across HeLa cells mainly. And this technology was pretty much developed by a very talented postdoc who was in my lab for about 10 years, a guy called Taka Nojima. So Taka basically developed a lot of the transcriptomic techniques, which my lab and many other labs are now using. But then the bioinformaticians, I had a very talented, really straight mathematician who switched to bioinformatics, who worked with Taka for a while, called Rita. And then the bioinformatic problem I had became possible because I set up a wonderful collaboration with somebody not far away from you guys in Lisbon, from you guys in Porto. And then this, of course, is Lisbon and Maria Carmo Fonseca. And so I've collaborated with Carmo Fonseca over the last 10 years. And we published a lot of, I think, quite nice papers together. And it's been very much down to the fantastic bioinformatics provided by a whole series of students working with Carmo. Thomas, Kenny, and now Rui have all been really instrumental in analyzing these complex data sets, often produced by Taka. And so we published a bunch of papers, and you'd be relieved to hear I'm not going to talk about them all, but they've all had gratifyingly quite high impact. And so they're all published either in Molecular Cell or in a few cases in some other journals. So I'm just going to highlight what we've learned from a few of these papers to give you some flavor of where we are today. So going back to long non-coding RNAs, where do they come from and how are they processed? Are they the same as messenger RNA? I mean, some people, when they first discovered long non-coding RNAs, just thought they were the same as messenger RNA, but they didn't code for anything. And that's not true. They have a lot of significant differences. So firstly, a lot of long non-coding RNAs actually are related to a nearby protein coding gene. So here's a simple cartoon of a protein coding gene with 3 exons. It could be alpha globin with the 2 introns in between, and the genes controlled by the promoter, the terminator, and usually multiple enhancers. It turns out that these regulatory elements are also the source of other transcripts. And so promoters go backwards to make antisense transcripts called prompts. You also get terminators initiating transcripts from backwards to make antisense coding transcripts. Then it turns out that enhancers invariably generate extra transcripts directly from the enhancer, behaving as if it's a promoter, which in a way it is. Really, enhancers are displaced promoters, if you like. So they make eRNAs. And then between protein coding genes, there's very often transcription units which don't code for anything that are called long intergenic non-coding RNAs. Okay, so to give you a for instance, so this is old data from a paper from Taka and Rita, where we just tried to map across the Hela cell transcriptome where the protein coding and non-coding transcripts were. And so here's a protein coding transcript called LBR. And what we've done here is we've analyzed the RNA that's in the chromatin fraction of Hela cells that makes LBR. We call it chromatin RNA seq. This is Illumina RNA sequencing, of course. And if you isolate the RNA that's in the nucleus, when you get rid of the chromatin, what you end up with is just the spliced transcripts. And so you just see exonic reads. But actually, if you look at the chromatin fraction, the reads are still peaking in the exon. So splicing is happening actually on chromatin as the transcript is being made. But then what's remarkable is that there's a link RNA next to LBR, which is transcribed in the chromatin fraction about the same level as the protein coding gene, but none of it gets into the nucleoplasm. It's all degraded actually as it's being made, we think. And then to give more examples, here's cyclin D, a regular protein coding gene, important in the cell cycle, obviously. This is the chromatin seq profile, transcripts all the way across introns and exons. And here's the nuclear fraction, which is just giving the spliced exons. So that's what regular protein coding genes will look like. But if you look at a very abundant long non-coding RNA, MALAT1, which is actually made in large amounts in the nucleus, it's 10 times more highly expressed than this cyclin D. But virtually all the transcripts are just on chromatin and degraded before they get to the nucleoplasm. And there's yet another example here, another link, which is about 50 kilobases in length. And all it does is code for 2 little mini pre-microRNAs, these little lines here. And the rest of it's all degraded on chromatin. So, but you do release 2 pre-microRNAs. So that gives you a sense of, so looking across the Hela cell transcriptome, and this is a study that Rita and Taka carried out, we published in Molecular Cell in 2007. What it really showed was that really most of these link RNAs are just chromatin associated and degraded as they're being made. And so we really think at least this class of long non-coding RNAs are very different to messenger RNAs, that they're rapidly degraded, they are spliced very inefficiently, and they don't have poly-A tails, they're usually poly-A minus. And so there are some functional long non-coding RNAs, and they seem and they need to somehow avoid being sort of turned over in the way regular long non-coding RNAs are. But Taka had this idea that maybe we could make Hela cells produce a lot more long non-coding RNAs than usual, and what would be the phenotype of those cells? And the way he did that was he knocked out a key elongation factor called SPT6. He depleted it using RNA interference. And if you get rid of SPT6, you have a remarkable effect on the Hela cell transcriptome, which is illustrated by one particular gene, which you'll all have heard of, called MYC. So MYC is a very important oncogene. It's associated with cancer, particularly in B cells and T cells. And essentially, if you get rid of SPT6, you don't change the pattern of transcription over the MYC gene. But if you look at the antisense promoter for MYC, you get this extraordinary long non-coding RNA being produced when you knock out SPT6, which is much longer and far more highly expressed than usual. And so that's really dangerous for the cell. You're making lots of extra transcripts that you shouldn't be making. And it turns out the way this works is that long non-coding RNAs, although they're transcribed by RNA polymerase II and they have SPT6 as an elongation factor, the SPT6 factor is involved in recruiting a complex called integrator, which is rather like the cleavage and poly A factor because it has an endonuclease in it which chops the transcript, but it doesn't add the poly A tail. So it tends to terminate LNC RNA transcription quite early. So you only make rather short RNAs of several, maybe 500 bases, no more. And so that keeps the LNC RNAs sort of close by to where they started. But if you get rid of SPT6, then lo and behold, you make these long non-coding RNAs like that MYC long non-coding RNA, which form loads of these R loops. And then you end up with the polymerase running all the way through. And actually, if this happens in the S phase of the cell cycle, the polymerase will now collide with the replisome, with the replication fork. And so you end up with collision, which can give you double-strand breaks and all sorts of sort of DNA and cellular stress. And so you end up with Hela cells going into senescence and generally dying ultimately. So making far too much and far too long LNC RNAs is very bad for the cell. So R loops, what are R loops? So RNA can base pair to the DNA template behind the polymerase as it transcribes through a gene. And we wondered whether R loops are quite widely present in the genome. So we used an antibody which picks up the RNA DNA hybrid. That's the R loop is actually the RNA DNA hybrid plus the single strand DNA. And so you can IP R loops using this antibody against the RNA DNA hybrid. And so you can then map these IP'd RNAs across the genome, and you can basically see lots of R loops over promoters and terminators in general, and elsewhere too. And then you can, if you overexpress RNase H in the cell you're studying, then you can get rid of these RNA DNA hybrids because RNase H will cut RNA when it's base paired to DNA. So we did a genome-wide analysis of R loops in Hela cells and what happens if you overexpress RNase H. And so here's a screenshot of a particular gene. It's mainly introns. It's got a few little small exons. So we mapped the R loops. There's a big R loop. There is an R loop near the promoter of this gene. There's also one over exon 2, in fact, for this gene. And then if you look at the chromatin sec profile across the regular gene, there are transcripts across the introns with big peaks over the exons because splicing is occurring as the gene is being transcribed. Here's the antisense transcript. If you overexpress RNH, the antisense transcript goes away. And if you just map the caps of the sense and the antisense transcript, which you could do by a cap-sec trick that we were using, then you get a lovely cap peak for the message and for the antisense transcript. And if you overexpress RNase H, you just lose the antisense cap transcript. So that tells you essentially what we concluded in this paper, which is that essentially over promoters, enhancers, and also the ends of genes, you get R loops forming, and this favors the formation of an antisense transcript, which then goes away when you overexpress RNase H. And so we think that actually a lot of these long non-coding RNAs that I've been talking about actually initiate transcription because of R loops. I don't know what the fraction is. It's probably well over half, maybe approaching 75% are R loop dependent. And so we think that LNC RNAs are made through R loops very often, and they terminate by this integrator mechanism to give you a poly A minus transcript. So really, it's very different to messenger RNA. And that leads me to the last point, Alex, you'll be happy to hear. So a paper we published actually earlier this year to show we've been publishing stuff even during this hideous pandemic. This is a study that's a combination of Taka's transcriptomic skills and the latest bioinformatician from Lisbon, a guy called Rui Sousa Luis. I may even be pronouncing his name right. That's remarkable. And so basically, what we've done here is, rather than doing ordinary chromatin RNA seq, Taka IP'd the polymerase in the chromatin fraction after he'd cut the chromatin with DNAs very extensively. And then by IP'ing the polymerase from the chromatin fraction, he could then isolate the nascent RNA that goes from the active site of the polymerase all the way back to the beginning of the transcript, at least in theory. And then these RNAs we subjected not to Illumina sequencing, but to this wonderful Oxford Nanopore sequencing technique, which allows you to get much longer reads of sequence, because Illumina sequencing gives you short reads that you have to line up along your gene. But in principle, Nanopore can give you much longer reads. And furthermore, it does no amplification. So you're looking at single molecules when you carry out Nanopore sequencing. And I won't show you much of the data because it was published in Molecular Cell earlier this year, but here's one of our favorite genes, beta-actin. So if you look at beta-actin and you look at the nanopore reads across it based on this technique, which we call point sec. No, actually point nano. In fact, we have different point techniques if you read the paper. There's 0.5, which is where we map 5' ends of this nascent transcript. Point sec is when we fragment the nascent transcript and do Illumina sequencing, and point nano is when you get the long single molecule reads. So with point nano, you can clearly see that across actin, there's a bunch of transcripts at the beginning. This is pausing of the polymerase at the beginning, probably. And then past the polyA site, which is the TES, you get a bunch of transcripts which are probably being chewed away from their 5' to 3', from their 5' ends by the exonuclease to promote termination down here, presumably. But then if you look in the body of the gene, you can see some cases which are red, which are being spliced as the polymerase. So if you look at the end of each of these reads, that corresponds to the polymerase, to the RNA in the active site of the polymerase, whilst upstream is the extent of the transcript going back to the cap site in these cases. And so you can see this particular transcript, the single molecule, has been spliced in all three of these introns, whilst in this case, the middle intron hasn't yet been spliced. So you get a beautiful dynamic picture. And I won't spend too much on this because obviously you need to look at the paper, but Rui was able to do some really fancy bioinformatics on this data set. And what we come up with is the fact that at least in the Hela cells, the protein coding genes, 40% of the introns are spliced when the polymerase is in the immediately downstream exon. So a lot of splicing is very fast, but about 60% occurs a bit slower when the polymerase has already got, say, into the next intron or maybe even further downstream. And so probably this is the type of delayed co-transcriptional splicing which is associated with alternative splicing to give you the different messenger RNAs to expand the proteome, as I mentioned. So there's lots more to do here, and there's lots more analysis, and I've only given you an outrageously sort of a minimal snapshot of the data, but that's it. That's all I'm going to show you today. I'm going to stop shortly. I'm just going to show you a picture of some of the lovely people who've been in my lab, and sadly, some of these guys have left my lab a while ago. So here's Rita, who's now working in a German genomics company, and Hannah's now got her own starting lab in King's London. Soma's now doing bioinformatics in Edinburgh. This is Ashish, Soma's husband, who's also got a position in Edinburgh, running his own lab. So it goes on. Claudia is now a junior PI at Babraham in Cambridge. This is Taka, just hiding behind Claudia here, who's now an associate professor in Kyushu University, Japan, happily. And this is well, I could go for everybody's name, but I haven't got time. But this is dear Kingo, who's back in Poznan, Poland, as some of you may know. Okay, so that's really it. So yes, so they gave me a nice little picture of prints of the soles of their feet in plaster of Paris, which they then coloured. And so this is called the proud feet. And I'm going to stop. Just I've said everything. So I'm still funded by the Wellcome Trust. I used to have an ERC advanced award. So we had quite a big group for a while. Now we just had the Wellcome Trust funding us. I'm stopping. Thank you, Alex. I think I've more or less obeyed the time slot, but who knows?
Thank you, Nick. An excellent talk as usual, and you kept on time. Excellent. Did I?
So how many minutes was that? I'm not good at judging my time, as you know.
No, you're perfectly on time. 40 minutes.
There you go. So I am capable of doing this because some of you who were amongst those listening will have heard me give talks where I ran over far too long, for which I do apologize. It's one of my dangerous tendencies, but I tried to control it because Alex seemed a bit fierce about 40 minutes, I have to say.
You have already 3 questions in the chat. So those of you who want to make questions, please write it down in the chat. First one, thank you, Nick. Great talk as usual. If you had to pick only one, what do you think was the most important breakthrough discovery in the RNA fields that occurred during your career?
Excluding the ones that my lab made, you mean?
Yes.
I don't know. What was the biggest breakthrough? I think probably still the realization that the protein coding genes have introns and that you get alternative splicing. I think that does deal with one of the big problems because, you know, with only 30,000 maximum protein coding genes, we had a bit of a problem. We know that there are 500,000 proteins, and so how on earth do you make them? So alternative splicing was a big discovery in solving a part of that paradox. It doesn't really solve it all though, because we don't understand how it's regulated, and there are many questions. But I think that alternative splicing, although that's not something in my lab discovered, I think that's a pretty big one, if you want my honest opinion. In terms of whether the Nobel prizes were awarded appropriately, yeah, I think in general they get the discoveries right. They don't always get the right people ascribed to the discoveries necessarily, but so clearly, I mean, there's a whole list of them. I mean, having the structures of RNA polymerase II were pretty important. That was Kornberg's discovery, although it should have arguably gone to Patrick Kramer as well as Kornberg. He did all the work, or some of the work. I don't know what the ratio was exactly. Then, of course, RNAi got prizes. That was important. Oh, CRISPR. We all love CRISPR, and there's no doubt that prize was appropriate. But about 20 people should have shared the prize, and they only gave it to 2 wonderful ladies who both deserved the prize, but not uniquely. So the list goes on. I don't know.
Okay, several questions, loads of questions. So one, what is the application of these discoveries, your discoveries? And if you have a theory, this come from Marco to explain the biological meaning of the paradox. And then we have a couple of questions from students.
Yeah.
So do you want to start by those? What is
well,
the application?
I mean, application. I always have to defend myself with the application questions, because I think understanding how things work is fundamentally important to the human race. I think we all seek to understand what's going on around us and how we are created and how things buzz. And I think, you know, without that knowledge, we wouldn't be able to defend ourselves from some of the terrible things that the world has to deal with at the moment. So clearly the pandemic was substantially helped by the discovery of RNA vaccines. And those RNA vaccines wouldn't have come without basic knowledge about PA signals and splicing and all the stuff that I was talking about today. So I think that's clearly an application. And indeed, Boris Johnson, our lovely Prime Minister, says how grateful he is to science for saving the day with the pandemic, and how he's going to commit an enormous amount of extra resource to funding basic science in the UK. But, you know, I don't believe a word that man says. Another question.
Do you have any advice for someone who wants to work in research?
Well,
it is a student for sure, or someone young.
Yeah, I think you should only go into research if you have a fascination to understand how things work. I don't think you go into research, you know, to have a very safe and well-financed career. I mean, it's pretty tough, because the problem with science is that you only really can justify your science if you discover something from time to time. But discovery is so much down to chance. I mean, it helps if you do lots of hard work and you're very clever, you have lots of clever ideas, but in general, it is a risky business. But so you have to be a bit crazy to do science for a whole career. But then, having done science for 50 years, I can't think of anything I'd like to do more. And I think it's a real privilege to be able to find out. I mean, what's amazing about science is that you do research, you discover something that nobody knew before. So everything that I've done throughout my career, at the time we did it, I think bits of what we did at least were totally new. and that's kind of cool.
Yeah. One of my students from last year, thank you for excellent talk. Do you feel ethics are evolving along with these discoveries, and which challenges are we still facing?
Well, I mean, I think what we have come to realize is how enormous our gaps in knowledge are. I mean, we have this, I mean, the transcriptome is a classic example. I mean, we just don't understand how you regulate gene expression in all the different cell types to generate the incredible beauty and complexity of an animal, or even, dare I say it, a human, with such a sort of limited amount of coding capacity and such sort of rather seemingly kind of uncontrolled and random processes. I mean, the whole process of transcription, it does seem as though RNA polymerase II doesn't really know what it's doing. It just sort of jumps onto any bit of chromatin it can get its teeth into and starts transcribing. Yeah. And somehow what it makes is sorted out into the good stuff and the stuff that needs to be got rid of. And how that's achieved is very hard to understand. I will have to come clean and tell you all that I don't have any strong religious belief. In fact, I'm a fairly strong agnostic, I suppose, if not atheist. But there's so much that we don't understand that there's plenty of room for any sort of help from a deity to sort of make sense of this all, because it's maybe beyond comprehension, some of the complexity we have to deal with. But yet the complexity is defined by the size of the human genome, which is really not so big. I mean, 1,000 million nucleotides, when you consider that only maximum 10% of it is doing anything, it doesn't seem enough information. So yeah, I think there are a few issues still. Don't worry.
lots of stuff to discover yet, right?
There certainly is. Yeah.
Claudius Unkel asks, what was the cellular phenotype of overexpressing long non-coding RNA of
well, basically the cell cycle stopped, went into G0, and then soon afterwards the cells just sort of died, basically. So the phenotype was pretty severe. But of course, we were looking at a very, I mean, I should immediately apologize for having worked on Hela cells for the whole of my career, which of course are very artificial cells. It'd be nice to do a lot of the experiments we've done on real primary cells. And of course, we dream about doing that, although that may not be in my time, sadly.
Well, we're doing that with Joanna Wilton. But
I know you are. I know you are. And clearly, we should. Clearly, you should. I mean, things may look very different in stem cells. Stem cells tend to have less repressed chromatin. There's a lot more wide open space in stem cell in stem cells, and so maybe there's a lot more fancy regulation going on. Then, of course, when you turn to the brain, it's clear that neurons do amazing gene expression tricks. And there's all sorts of fancy stuff like sort of single amino acid exons, and you get this extraordinary business of you have a neuron which has its nucleus in the brain, and then the synapse is in the big toe, and somehow you get communication between the synapse and the nucleus to produce the right message. You know, there are extraordinary questions that will be a lot of fun to sort out. So I wouldn't worry as a student about not having enough questions to get your teeth into. The problem is to think of doing something sensible and something focus on mechanism rather than just collecting lots and lots of data. The problem now is we have the capacity to collect so much data that you get overwhelmed by it. And so you see so many papers where really there's vast amounts of data, but there's often not a lot of biology.
That's absolutely right. And that goes into the next question. Thank you for the great talk. What do you think will be the next big challenges for bioinformatics?
Well, I think there is one thing we really do need badly in bioinformatics, which is some way of easily dividing genes up into different categories of gene expression mechanisms. Because I mean, there's a tendency to put, to stick all of your genes into one metagene profile. And the problem with that is you then average everything out to nothing very much. So what you really want to do is to develop pipelines where you could really focus on the set of genes that have this particular trick and another set which have that trick. Yeah. To try and really work out simple bioinformatic ways of sorting out genes into different categories of mechanisms of gene expression, which is sort of happening, but I think it needs to be made more fluent and straightforward.
Not all genes behave the same manner.
All genes use to some extent the same gene expression apparatus, but they clearly do so in a manner which is quite gene or cell or whatever specific, and that's where we need bioinformatics. So of course, one of the big areas of gene regulation that's being investigated is looking at single cells. But the problem with single cells is you never get enough RNA to really do the fancy kind of sort of point-sec analysis I described at the end of my talk. And so we're going to have to somehow improve sensitivity in order to really get the kind of nitty-gritty detail you need, but on a single cell. But then, of course, single cells, of course, in multicellular mammals don't exist. I mean, they're all part of what they do in some cases, but in general, they're parts of tissue. And so, of course, it is the combination of all the liver cells that give you the proper liver phenotype. And individual hepatocytes may be misleading. You have to look at a whole set of them. So you probably need to do sort of transcriptomic analysis on 1,000 hepatocytes from one particular lobe of a liver to really get the full picture of how the liver is, of how gene expression is regulated in that tissue. And of course, when you go to the brain, it's far more complex than the liver. You've got all the different little bits of the brain, you know, what's going on? Well, like there's that bit of the brain which shrinks when you've had COVID and you can't smell and taste anymore, right? So it'd be quite nice to know what the cells are inside there and why that whole bulb is shrinking in size and what's actually happening. I mean, there are zillions of fascinating questions.
And again, in the brain, you have so many different types of cells with so many different functions. So you can't. And
the brain clearly does fancy things. There's lots of there tend to be much longer transcripts in the brain, and there's also this concept of splicing. Alternative splicing is normally happening in cis on one transcription unit, but there is a possibility of trans-splicing, of splicing a bit of one primary transcript to another one, to give you an even bigger mixing and matching of exons. And there's evidence that might happen in the brain. You know, so there's a lot of stuff.
I have 3 or 4 more questions. Enrico, the question is about the gigantic poly-A complex. Why so many protein factors are required? What the proportional is definitely required?
Well, I think in mammalian cleavage and polyadenylation, we can say categorically that you need 20 polypeptides. There's probably another 30 in the wings, which are sort of in some of the complexes. It's probably the case that not all the polyaosome complexes are identical. There's probably subcomplexes which have a particular function in different cell types. I mean, there is one that's well known. Histone messenger RNA is processed by a different mechanism, doesn't put a poly-A tail on the end, and a big chunk of the cleavage polyaosome is involved in histone mRNA processing, but it's it's got some different components. So there's all sorts of extra complexity here. And of course, how these complexes come together is another major question that we're only beginning to get a few clues about. Of course, one of the really big clues, which a lot of people are excited about, is the idea that you can get association of proteins and indeed RNA loosely by forming these condensates. And then these condensates basically give you a concentration of a subset of factors, which then allow a tighter interaction of a complex within the condensate.
Phase separation.
The phase separation. Yeah, there are all sorts of names. I rather like the name blob, but I suspect that's not very scientific. but yes, it's phase separated. Phase separated. There are all these structures in the nucleus which seem to be held together, but yet there's no membrane around them. So they're held together by these condensate sort of factors. But this is an exciting area, and there's undoubtedly condensates associated with specific loci and particular genes. And I think these chromatin-associated long non-coding RNAs may well be playing a role there. In fact, a good example, which is beginning to be understood, is XIS, which is an important long non-coding RNA, which does have a function, a very important function, and is clearly associated with some sort of condensate tendency formation. And yeah, there's lots of stuff.
How is SPT6 integrator restricted to long non-coding RNAs from the Oliver?
It's not. So basically, SPT6 is associated with all POL2 transcription, and integrator, we don't quite understand what the molecular basis for integrator association with chromatin is, but if clearly, if you get rid of spt6, you lose integrator. That's what we know. We don't know exactly what the molecular contacts are, but integrator seems to be associated particularly with either the promoter region of protein coding genes. It's also sitting on enhancers, and we think that its function is to terminate transcription prematurely. I think quite a lot of transcription never gets anywhere. It starts transcribing and then it's terminated after a few hundred nucleotides. And that's probably happening in protein coding genes to a large extent, but it's probably the main mechanism that operates on long non-coding RNA. So long non-coding RNA are mainly the short transcripts, which are kept short by the integrator. And the reason you need to keep them short is if you don't, then you'll make these long transcripts, which may give you, big problems in the cell cycle and whatever. I mean, in fact, MALAT1, which I cited as a very highly expressed long non-coding RNA, the reason that's known about is it's overexpressed in various forms of cancer. So overexpression of long non-coding RNA is clearly very deleterious. But there's, one story isn't going to account for everything. There's going to be differences between different situations for sure.
From Joanna Wilton.
Hi, Joanna.
Hi, Nick. Do you think that tar loops may pose an evolutionary advantage? Thanks again for the great talk.
Well, they do promote double-strand breaks in many situations, and that's normally bad news for the cell. But of course, you well know, Joanna, that double-strand breaks are critical for antibody gene function. You get class switching and VDJ recombination, and then the T-cell receptor also does double-strand breaks and recombination to generate diversity. So clearly, evolution has put these double-strand breaks to big use, at least in immunological cells. But it's dangerous. As soon as you have a double-strand break, you can get sort of a fused sort of incorrect sort of chromosome, which can be very deleterious. So I think I would have said overall R loops are more of a nuisance than a benefit, but maybe they have an evolutionary benefit. And clearly, as promoters for non-coding RNAs, I mean, most non-coding RNAs probably aren't doing very much, but there are some which have been evolved into something really useful, and that may have begun with an R loop promoter. So yeah, so I mean, you can argue the case either way, can't you?
Yeah. From Nike, nice talk. Is there something in the structure or coating of the long non-coding RNA compared to mRNA to make them unstable and degraded as they are transcribed?
Well, we don't really know the answer to that. I mean, actually, what I could do is I can tout a review. So Taka and I have just written a review for Nature Reviews in Molecular and Cell Biology, and this is exactly what we talk about in this review. But unfortunately, it's not yet published, but it will be published by the end of the year. So you can have a read of that, and we try and describe what's known about the non-coding RNA, long non-coding RNA termination in particular. So there seem to be, it's not just the integrator that terminates transcription in long non-coding RNAs. There's another complex involving a protein called WDR22. I may not have got that right, which I've actually given it the name in my review as the restrictor complex, which is also involved in long non-coding RNA termination. So there's a lot of complexity here which we don't really understand yet. Lots to find out.
Lots to find out to the younger students. I don't have any more questions in the chat. Is there anyone else with a burning question? If not, I think we can finish the session.
Wow.
Yeah, thank you very much. It was a great talk. Thank you for coming virtually. Hopefully we'll have you soon. And thank you for the audience for the great questions. Do not miss the next webinar next week. So we'll have every Thursday at 5 GMT or Lisbon time or whatever. Thank you, Nick. It was great to have you here.
Well, it's been a pleasure, and I'm pleased I managed to more or less keep to time, which is
yes,
unusual for me.
You really behaved really well, so well done. And you made, you know, an overview of everything since the discovery of molecular biology until hardcore transcriptomics. So I think the students had a view of the field from the very beginning. So that was very nice.
Yeah, but I think it is the case that it would be nice if students today had some appreciation of these 50 years, because in a way the field you see it developing. And I think if you just join the field at the present day, you lose a lot of the information about how everything was built up and how the ideas developed over time, and actually how much we got completely right in the early days. I mean, it's actually very gratifying that you know, I found a few messenger RNAs with the poly-A signal, and lo and behold, it's everywhere. And it is the case that although it's important to do genome-wide analysis, if you analyze one particular gene and work out exactly how it's regulated, that's not just going to be true for that one gene. It's going to be true for a whole family of genes, because there's only one set of apparatus, and so every gene can't have a totally different way of expressing itself. There has to be subfamilies, which is why I go back to what we need the bioinformaticians to do. They need to work out clever ways of finding the subfamilies, so that rather than getting a metagene of everything, you divide up the families of genes into maybe 50 different families, and each family you can maybe take to pieces and get more mechanistic information about them. That's kind of what I think about. But yeah, it needs some very fancy. It needs some fancy computer biology, really.
I have one last question from Maria, one of my veterinary students. How do you feel about those early days discoveries? I'm still stunned by them, but I'm only a student. How do you feel about those early days? You just said, really.
How do I feel about them? Well, I mean, we had an enormous feeling of frustration and confusion that we couldn't do anything, we didn't know anything. But then, of course, that led us to being able to do things and knowing a bit more. So it's been very satisfying, really. I think I've been very privileged to be in this field from its onset to 50 years on. And there's loads more to do, but I probably can't. I could do a bit more myself, but you know, I have to stop eventually. So there's plenty of stuff for you youngsters to do. So you should go for it, but only do it if you really have a fascination and a craving to understand how things work. I mean, if you just want to be a scientist because it's a safe career, then there are better safe careers by a long way. But it is fascinating. I mean, the beauty of gene expression is overwhelming, and I hope that's come across from my talk, that it is incredible that we have what seems to be complete chaos, and yet from that you can produce something as beautiful as a human baby. I mean, how is that possible? I mean, it's staggering, really. And then it develops into something a bit more ugly as it gets older, I suppose, depending on the baby.
Not so cute. Okay. Thank you again, and see you next week. Well, not you, Nick.
No, sadly not, Alec. But I'll see you soon, I hope.
Yeah.
Okay. Bye everybody in Porto and everywhere else around the world, wherever your computer is connected from.
You have lots of people around the world. You do. You do.
Okay.
I've seen them on the chat and on the screen. So yeah, well done, Nick.
Okay.
In fact, loads of people around the world. They're following you.
Bye everybody.
Bye-bye.
Good night.
Bye.